Good morning, students. Further, we will study about sexual reproduction in Riccia. Distribution of sex organs. The sex organs are developed on the thallus lobe, which are not in any way specialized for the purpose. They are developed in a line extending back from the growing point. Generally, they lie in the dorsal furrow or groove, sank deeply each in a separate cavity. They are developed in an acropetal order. The younger ones are thus near the tip or near the growing point, and the older are away from it. The antheridia and archegonia in some species are developed on the same thallus. Some species are known as monoecious. In other, the two kinds of sex organs are developed on different thalli. They are referred to as dioecious. Riccia crystallina and melanospora, Riccia gigantic and Riccia parthenocotensis are monoecious, while Riccia discolor and frosti, Riccia frosti are dioecious. In monoecious form, the antheridia are formed first and archegonia later. In a few cases, they are formed successively, a few antheridia and a few archegonia. The sex organ arise singly and each lie in a separate pit. The site of a sunken antheridium is usually marked by a tiny tower-like projection of the surrounding tissues. The thallus of Riccia, as it bears the sex organ, is known as the gametophytic plant. The sperm and the egg are the last structure developed during the gametophytic phase. Structure and development of sex organ. Antheridium. Antheridium, the structure. The mature antheridium is an elongated structure. It is consists of an ovoid pear-shaped body seated on a short few cell stalk. It stands in a deep pit in the anterior chamber and is attached to its bottom by means of multicellular stalk. Each antheridial chamber open at the upper surface of the thallus by a narrow pore called an ostiole. The body of antheridium has an outer jacket layer of sterile cell. It is called the antheridial wall. The antheridial wall is protective in function and 1 cell in thickness. It encloses a mass of small fertile cubical cell called the androcyte mother cell. Each androcyte mother cell has a dense cytoplasm and relatively larger nucleus. It divides diagonally to form two sperm cells known as spermatids or the anthrocyte. The protoplast of each anthrocyte get metamorphosis in a single sperm. In this way, several hundreds of flagellated sperms are produced in each antheridium. Each sperm is a minute cylinder structure, curved structure. It is furnished with a pair of whipless flagella at its anterior end. The body consists of an elongated nucleus. The portion of the cytoplasm not utilized in the formation of flagella remain attached to the posterior end of the body of the sperm as a tiny vesicle. The sperm do not leave the antheridium until enough moisture is present to permit them to swim about. Dehiscence. Presence of moisture is the essential for the dehiscence of mature antheridium, in which the wall of the anthrocyte or the spermatid have dissolved and the sperm lie free in the viscous fluid in the cavity of the antheridium surrounded by the jacket layer or antheridial wall. Water enters the ostiole and finds its way into the antheridial chamber. The cell of the apex of the antheridium absorb this water by imbibition. They get softened and eventually disintegrate to form a distal pore. The sperm may be shot with a considerable force or extruded slowly in a viscous mass through the pore. Eventually, they escape through the narrow canal of the antheridial chamber to the upper surface of the thallus. Here they swim freely in a thin film of water in the dorsal furrow. Thank you.